Thanks, David, and thanks for the opportunity. Obviously, a few months ago, I decided to speak at the meeting. A few weeks ago, sent in the information that's in your brochure, and it's already out of date, and I have to say pleasingly out of date, because I had a look at the share price as it's reflected in the book, and things have happened since it was printed. And that's going to be part of the story I'm going to tell you. In fact, most of the story I'm going to tell you today. So for those of you unfamiliar with Actinogen, we licensed in technology from Edinburgh University 5 years ago. It's a family of drugs that are designed to get into the brain to inhibit a particular enzyme, the 11-beta-HSD1 enzyme, that occurs in all cells, but primarily in the brain, in the frontal cortex and hippocampus. And this enzyme produces cortisol. This is what it's designed to do. So our drug inhibits the enzyme and so inhibits cortisol production. And what is recognized, is that persistently elevated cortisol, which is the stress hormone. Persistently elevated cortisol is associated with the development and the progression of Alzheimer's disease, and in fact is associated with cognitive impairment across a number of other diseases: diabetes, schizophrenia, depression, Parkinson's disease, PTSD, epilepsy, a whole range of them. So by inhibiting that enzyme, bringing down cortisol, the hypothesis is that we can make an improvement either on cognitive impairment in those diseases or on Alzheimer's itself. And that's the hypothesis that underlies the development of our drug. The work has been underway at Edinburgh, for some 10 years before we licensed the family of drugs 5 years ago. We've since then done 4 capital raises, raised 35 million, continued the development of the drug. The drug's been in humans now for some time. Over 200 patients have taken our drug. No safety problems. We've got good IP protection for our drug. And the principle underlying the cortisol inhibition, I think, has now been proven by the data that we've got. So everything is looking very rosy for us to now move into the next phase to demonstrate that our drug improves Alzheimer's or can provide benefit to the patient with Alzheimer's disease or cognitive impairment associated with any of the other conditions that I've alluded to. But one of the key points in our development program was the initiation and completion of a major international Alzheimer's trial, mild Alzheimer's disease, that we initiated some 3 years ago and read out in May this year. It was a 12-week trial, 186 patients, run in Australia, the UK, and the US, and in fact was the largest international Alzheimer's trial ever run by a biotech company in Australia. Importantly, and this is an important point I want you to remember, the patients were dosed at 10 milligrams a day. Now, on the available data we had back then, that was the optimum dose to use. So 10 milligrams a day in Alzheimer's disease, that was the trial. As reported in May this year, while we showed that our drug got into the brain, inhibited cortisol, and was safe at 10 milligrams a day, we didn't show the efficacy signals, the statistical improvement in cognition that we were looking for from this trial. It was concerning because until this point in time, the drug had behaved exactly as predicted from all of our modeling. And one of the considerations was, did we have the dose right? Now, as I say, the assumptions we made back some 3 years ago was that 10 milligrams was the correct dose, but we recognized the potential that we may not have the dose right, and so initiated a whole lot of additional studies in May 18, one of which was a higher dose safety study that included a cognitive endpoint. And that's the study that reported out 3 weeks ago, and produced such spectacular results. But alongside of that safety study, we initiated a target occupancy study and a number of high-dose toxicology studies to round out the data set that we needed for our drug to prepare us for the next phase of research. So, in May this year, while we showed that our drug worked at inhibiting cortisol, unfortunately, we didn't hit the endpoints, and our share price tanked. And that's what you see in the graphic in the booklet that you have. But I'm pleased to say things have turned around since then, and it's all due to the higher dose safety study that we initiated in April last year. So this is the portfolio of studies that we have ongoing right now. Right at the top, the Alzheimer's trial that I've told you has reported out. We're just doing final analysis on all of the data, looking for trends, looking for indications, but that study is complete. We have a phase 1 study and homogeneous binding study, an in vitro homogeneous binding study, and a phase 1 human study ongoing at the moment, target occupancy study. We have the higher dose safety study, the Xanamem study. This is a safety study run at 20 milligrams a day. Now remember, the Alzheimer's trial, the Xanadu trial, was 10 milligrams a day. So this was a safety study that we initiated at 20 milligrams a day to get safety data that we would need for increased doses in the future. But as I say, along the way, we included efficacy data with that study. We also have a number of toxicology studies ongoing to allow us to run longer-term studies in the future, and new indications have been identified, and the primary ones that we are looking at include cognitive impairment in schizophrenia and bipolar disease, two hugely significant unmet medical needs and big market opportunities alongside of Alzheimer's disease that we recognize present a great opportunity for our drug. But really the good news that I want to share today is the Xanamem trial, the higher dose safety trial. As I said, run at 20 milligrams a day, but this is in a healthy elderly population. So people without a cognitive impairment, generally a healthy population, elderly between 50 and 75, dosed at 20 milligrams a day for 12 weeks, exactly the same as with the Alzheimer's trial. Only 42 subjects, though, a small trial in comparison to the Alzheimer's trial. But importantly, 20 milligrams a day, placebo-controlled, so 30 of them on the active drug, 12 on the placebo, but with a cognition module added to the trial. The reason I added that to the trial is some data generated way back in the beginning of the development program at Edinburgh University showed that if you inhibit the enzyme, the 11-beta-HSD1 enzyme, in elderly people, healthy elderly people, you can produce a cognitive benefit, a cognitive response. We decided we were going to replicate that data with our drug, and that's why we included a cognitive endpoint in what was primarily a safety study. Now, this is a lot of data here. I'll walk you through it slowly, but don't be concerned about the data. I'll give you the key outtakes. But this is the data from the study. The cognitive battery was run by Cogstate, a company very, very familiar to the whole pharma industry involved in cognition research. 6 domains to their test, and you can see the p-value and the treatment effect in the different columns. P-value in the middle column and the treatment effect on the right-hand column. The p-value, any value 0.05 or below, is a statistically significant p-value. but a 0.09 against the detection test, the one looking at psychomotor function, in a study of 30 patients is considered statistically significant. So really, we're looking at 3 statistically significant endpoints in a patient study of 30 active patients. A very remarkable, very telling result by any measure, and one that took us by surprise. As you can see, clearly we sent in data reflecting a share price before we knew this was happening. The working memory at the top, a p-value of less than 0.01, highly statistically significant. And if you look along at the treatment effect, this is another way of demonstrating the significance of the results, that the effect of this treatment occurred at week 4 and continued out to week 12. So it occurred early and continued right through to the end of treatment. Just to reflect on this treatment effect, it's a measure of the actual significance of the results. How different were the results seen in the active versus the placebo group? And any figure above 0.5 is considered a good treatment effect. Above 0.8 is considered a strong treatment effect. So this is remarkably significant data, considering that it was just a 30-patient study. Now, just to put this, to provide a bit of context to this, Aricept, the Alzheimer's drug, the market-leading Alzheimer's drug, from Pfizer. In fact, I was involved with developing it 25 years ago at Pfizer. Aricept produced a treatment effect of 0.2. Now, in no way am I trying to compare the two. I'm just providing that as a reference point so that you understand that if a drug can be the leading drug in Alzheimer's disease for 25 years, generate annual sales of 5.5 billion at peak. If that can be registered with a treatment effect of 0.2, it says something very significant about these results. Anybody that has seen these has been suitably impressed, clearly, as have big pharma companies that have seen this. So very pleasing results, and results that clearly indicate to us that our drug works. Looking at the same results or similar results, but looking at the data graphically, you can see again a very rapid response and a clear statistical significance across the treatment time period. Just to be clear, the treatment stopped at week 12. Between week 12 and week 16 was an off-treatment safety evaluation time period. But again, a very clear representation of the effectiveness of our drug. And the final slide I want to show you on this trial was the effect on cortisol. Here you can see statistically significant decrease in cortisol in this patient population. Now, for those of you who may be concerned, did this reflect in any safety signals in the study? No, the safety was fine. So this was just simply a decrease in the cortisol, but a decrease without affecting the safety of the patient, which is clearly paramount. So what we have is clear evidence that our drug gets into the brain, it inhibits the enzyme, it decreases cortisol, and it improves cognition, which is exactly what the drug is designed to do, exactly what all of our animal data indicated, and really sets us up beautifully for the next round of studies. The final bit of data that I just want to share with you is another study that we have ongoing still right now, being run actually at the Austin Hospital here in Melbourne by Professors Chris Rowe and Victor Villamagna. This is a phase 1 target occupancy study where a radio-labeled drug is given to the patient, and the binding of our drug is demonstrated. Now, It's very technical, but just to demonstrate visually what we've achieved just in one of our patients. We have some 20 patients on the trial. Just in one of our patients, the top line, the colored line, you see it referenced as baseline. That is where the radio-labeled drug is given to the patient, and a special imaging technology, a PET image, is taken of the brain, and you can see the radio-labeled drug bound very elegantly to the enzyme throughout the brain. The second part to the study is then that same patient is given our drug, in this case, Xanamem 20 milligrams, is given our drug for 7 days and then redosed with the radio-labeled tracer. And as you can see, there's very little binding of the tracer in the lower set of images, reflecting the fact that our drug very effectively takes up all the binding sites of the enzyme in the brain. So here we have further proof of our drug getting into the brain and effectively binding to the enzyme. When it's evaluated mathematically, we achieve between 50 and 85% binding, which is exactly what we wanted to achieve. Now, we're doing this at different doses of our drug to be sure that we can define exactly what is the optimum dose for our drug going forward. But all of that data will be available before the end of the year, and we'll be ready to initiate the new round of studies early in the new year with this important data on hand. The toxicology studies I've already alluded to, so I won't mention those anymore. So really, the key outcomes that I want to leave you with is, after the disappointment of the Alzheimer's trial in May and the tanking of the share price, the piece of that graphic that's not in your handout is what happened to the share price after October the 1st. When we announced this great data, we're back up to where we were before, which is exactly where we hoped to be all along. And now we're ready. In fact, we're even more prepared than we expected to be to go forward into the next set of studies. So all our data shows that between 10 and 30 milligrams of our drug achieves good target occupancy of the enzyme in the brain. They inhibit cortisol in the brain and achieves a statistical significance of decreasing cortisol in the periphery without safety problems for 12 weeks of therapy. But most importantly, and this is why the drug was developed, we showed a statistically significant cognitive improvement in these healthy volunteers after 12 weeks therapy. And this result occurred very rapidly after 4 weeks of treatment and continued out to the end of therapy. So now we're ready to move forward into the next round of studies, which clearly are going to involve Alzheimer's studies. We are busy speaking to our advisory boards, our key opinion leaders right now, and designing these studies to initiate next year. We're hoping to initiate 3 or 4 studies next year, clearly in Alzheimer's disease, clearly also in schizophrenia, cognitive impairment in schizophrenia and bipolar being our second strategic directions. And then we've been approached actually by some academic units overseas who've seen our data, very excited with the data, and very keen on running studies in particular therapeutic areas that they're interested in that are low down on our strategic list, but equally very credible therapeutic opportunities for our drug. They're very keen on running those trials. We've said we'll make the drug available to you if you raise the capital and run the studies and make the data available to us. Those negotiations are ongoing, but I'm very hopeful that they will generate some additional studies as well. So this time next year, I'm hopeful that I'm standing here explaining to you the progress we're making and demonstrating the 3, 4, 5 studies ongoing across a number of therapeutic areas with our drug, and leading up to obviously the exciting readout of our drug in therapeutic areas related to raised cortisol and cognitive impairment. Thank you for listening. Thank you.
Bill, I've got a question for you. It's to just take us back a little bit more to the underlying hypothesis to do with cortisol, why cortisol matters. But can you also take us back to the ABLE study and some really interesting findings that came out of that study? Take us back to the ABLE study, because that's an Australian study, a long-term study into ageing, and with input from many researchers in Australia, but it came up with a very interesting finding about cortisol.
Yeah, so let me speak to the ABLE study first. This is as David says, a very interesting Australian study, a prospective study. So and it's funded by the CSIRO, so completely independent of us, run nationally, primarily in Melbourne and West Australia, but run nationally. And it's taken a healthy elderly population and followed them prospectively over 10 years and taken serial brain scans, blood tests, cognition tests over those 10 years and produced now well over 200 peer-reviewed papers, presentations, and it's all available on their website. So a very rich data source, internationally rich data source, producing extraordinarily useful data, looking at normal brain aging. One of the papers they produced about 18 months ago, a year ago, showed that same population but looked at cortisol. So this is an elderly, cognitively normal population with or without raised cortisol. And what they showed is if you're cognitively normal and elderly, and we're talking of people in their 50s and above, so, not 75, if you're cognitively normal and elderly, and you've got a persistently raised cortisol, you're at much higher risk of getting Alzheimer's disease. And the higher that cortisol is, the more aggressive the Alzheimer's disease. And their conclusion is that we need to find ways of inhibiting cortisol production to try and slow down or even stop the development of Alzheimer's disease. Now, where our data offers an enormous possibility is that we have that potential in that healthy elderly population without demonstrable Alzheimer's disease, but with raised cortisol. We have that potential to be a preventative, but that's one step at a time. What we need to do, first of all, is demonstrate that our drug works in the treatment of Alzheimer's disease. If it does, it offers magnificent opportunity to be used earlier because, as we've seen, in a healthy elderly population, we already have that data that it produces a cognitive signal. Very exciting stuff. And the possibilities that flow from it are enormous.
Another question then?
The overseas academic studies, are they on diseases that are very big in terms of population numbers?
Yes and no. is all I can say. They're in different disease populations. The one is a very, very big disease population. The other one is more of an orphan indication.
Okay. And will you have the IP around those after the studies?
Yes, absolutely. Absolutely. We're not licensing out the drug. We're retaining the IP. We're simply making the drug available to them for them in their academic interest to work with our drug in their particular therapeutic areas of interest.
Just one final one here.
Very impressive with this achievement so far. I would like to know, you have another 3 and 4 studies next year. When these results coming out and when gonna goes to commercialization. And
Yeah. Okay. So clearly, this data we received now, as pleasing as it is, took us a bit by surprise. So in the 3 weeks since we received this data and really understood the true significance of it, in that time we've been aggressively planning the next phase of studies. So there's a lot of detail around the size of studies, what the design will be, the number of patients, where we will run the studies. All of that detail has still to be worked out. But I have a personal goal that I would like to be able to make an announcement by the end of the year around most, if not all of those studies. So the goal is within the next 2 months that I'll be able to provide more information around the studies that we plan to run next year. Actually, how long they will take, again, that all depends on the size of study. But I would say what we have here in a 42-patient study is one model that we could use. Now, clearly the statisticians will look at that and say, statistically, that's not a powered study. You can't run that study. We need to do a properly powered study that might be 150 patients going forward. Equally, what we've shown here is that a 42-patient study can be a properly powered study. So we are going to need to balance those two priorities. And it may be that we run both shorter and longer studies in parallel so that we can generate both statistically powered, properly powered studies at the same time as generating very rapid data in smaller patient populations. I hope that answers your question. At this point in time, we don't have certainty around that.
Okay. Thank you.
Cheers, David. Thank you. Thanks very much.